Swayam Prabha. Digital India. Educated India.
So, we call this, we were looking at the fiber arrangements in the muscles, and the two broad classification are the parallel and the penniform or pennate arrangement, also known as penniform arrangements. And these, when we say fibers at this sort of macroscopic level, when we are talking about the muscle, they are actually called fascicles. Okay, we will see why when we look at the structure of the muscle fiber, because we will go into the fascicles, then into that, etc. So, but when you are talking about the arrangements and we say muscle fibers, we know what it means. Okay, we are talking about. So, if you look at the tendon, so the muscle fiber is what is between one tendon and the other. Okay, and it is that configuration that we talk about in this parallel arrangement or the pennate arrangement. So, if you look at the parallel arrangement as we saw here, this would be your tendon. Okay, you have the tendon, and then you have, so you see here, actually you see the white tendon here, right. So, those are the tendons, and then between those you have the muscle fibers running parallelly. Okay, so, this is the parallel arrangement. You have tendon at either end, and between them the muscle fibers are parallel. Now, the penniform or the feather like arrangement is, I said there are three. You can have a unipennate arrangement, where essentially you may have a tendon like this, a tendon like this. Oops, I should turn off the touch on this. Okay, and then the muscle fibers actually run at an angle between the two tendons to the direction of pull. So, the tendon will be pulling on, whatever it is, the bone that it is pulling on, it will be attached to the bone here, okay, and here. Okay. So, the tendon is pulling like that, but the muscle fibers are at an angle to the tendon. So, the unipennate would be something like this. So, that is, so it may not be very evident in this figure. You see here both the fusiform and the unipennate look more or less the same, but this is what is happening with the unipennate. So, when the muscle fibers contract, these two, they are contracting in this direction, okay, but the force that is being applied to the bone is at an angle to that. Okay. So, it is not the full, so these muscle fibers are generally shorter, but you can pack a lot more muscle fibers in that muscle. Okay. And so, there the amount of force that they can generate is much higher. So, in the case of the parallel arrangement, you get a lot of contraction, you get a lot of displacement. Okay. With the penniform arrangement, you do not get as much displacement, but you can get larger forces, although it is only a portion of the force that is being applied along the tendon, it is still larger forces are possible, because you are trying to sort of, the body is trying to maximize, depending on the configuration, maximize the area for that particular muscle. Yes.
Like tendons are a part of bone or they are separate
They are separate. They are a connective tissue. So their composition, they are not part of bone. They are what connect the muscle to the bone. So their composition is different from the muscle composition. Bone composition is different. Tendons are different. Ligaments are close to tendons, but they are still different. So if you look at their chemical composition, everything will be, even the structures will be slightly different.
Tendons, do they have flexibility?
Yes, they do have flexibility because they move around joints. Okay, they can bend. They are like flexible members. Okay, and they are also elastic to some extent, not as much as muscle, but they can undergo slight strains. We'll talk about tendons a little bit more as we move along. But yes, they are elastic, they are non-linear. So they actually function more like non-linear springs in the body. Okay. Your muscles are your actuators, similar to your not exactly motors, but more like hydraulic actuators. Okay, but they can only contract, okay, and apply tension. Okay. So this is the unipennate. The bipennate, similarly, you can probably look up some examples like the rectus femoris, but they may have, so these tendons may have like a central, the tendon may have like a central thing from which you have, and then you have the other tendon perhaps, and the muscle fibers between them could be like this. Okay, something like that. That would be like a feather, and you have the tendon on either side. So the tendon may be something like this, then like. Okay, so that's your bipennate, multipennate muscles. More branches than this. You know, if this tendon instead of just one central, it has multiple ones, then you have like a multipennate muscle fiber arrangement. Okay. So you can see here, this is a, you can see the black parts are the tendon, it shows here. I presume that is what the person who has drawn this is trying to say here also, but essentially you look at a muscle fiber, it has to connect from one tendon to another. Okay, that is the way you identify the arrangement. You see how it is connected to the other tendon. So this is your, and here if you look at it, you can see that if this is the direction of pull, okay, then this blue thing is your anatomical cross sectional area, okay, but it is not really, let me just, so if you take a slice of the muscle perpendicular to the direction of pull, then that is your anatomical cross sectional area. I should actually draw this the other way because no, the muscle is subject to, the muscle exerts tension. So, this is going to be pulled this way, right, the force on the muscle is there. Now, the green, if you take a cross section perpendicular to the fibers, that is really the measure of the force production of the muscle. Okay. So, the green thing is what is your PCSA. This is your physiological cross sectional area. Okay. So, in a multipennate muscle, the sum of all these areas gives you a measure of the total force that muscle can produce. Whether that is all that force will not be along the line of action of your tendon, but you can also see in the peniform muscles the fiber lengths are shorter than the tendon or what is known as the whole muscle length. Okay. So, they are shorter than the muscle length. So, in the peniform you have fiber length less than the muscle length. Now, this is defined as the physiological cross section area. I'll give you the del. Yes.
Fiber is something that makes the muscles, right?
Yeah.
So, if fiber is constituting the muscles, then it should be equivalent to muscle length as well.
So, we will look at how the muscle is, what the muscle structure is. Okay. So, when we say muscle length, it is from the end to where the tendon, from end of one tendon to this, that is your muscle length. So, this to this is my muscle length. Okay. That the distance between that is my muscle length, but you can see that the fiber length is less than the muscle length. In the parallel muscle, in the parallel arrangement, they are more or less equal. Okay, they are the muscle length is equal to the fiber length in most cases. Here the fiber length is generally less than the muscle length. So, with the peniform arrangement, let me make another copy also. In the peniform arrangement, you can see that the force, the muscle force, let us call it Fm, okay, is this, and the force that is transmitted to the tendon is Ft. Because this is at an angle theta, the theta is called the pennation angle, and the force that is transmitted to the tendon equals that times cos theta. Okay. So, the definition of PCSA is the sum total of all the cross sections of the fibers in the plane, perpendicular to the fiber direction. So, because the fiber length is smaller than the muscle length, the you have smaller range of movement, but larger force production in the pennate type of arrangement. Okay. So, now we will look at, so this is like I said, we first started off with okay, muscles are in compartments, then an individual muscle, I say okay, I can see fibers in the muscle, I can look at that arrangement. Now, let us go in a bit into the structure of the muscle. So, if you look at the muscle, it is covered by, so it has these fascicles, right. Each muscle may have, so these fascicles are like fiber bundles, okay. Fascicles are so each muscle will have hundreds to thousands of these fiber bundles, lots of bundles in one muscle, okay. And then the fascicle itself will have hundreds of fibers, will have several fibers inside. So, it is like tubes within tubes within tubes, bundles within bundles within bundles, okay. So, you have, let me tell you, so the outer fibrous layer, this is called the epimysium, and this is what transfers the tension to the tendon, okay. So, this epimysium contains all the bundles, fascicles, okay. Then you look at, so you have several of thousands of these fascicles inside the epimysium. Then each fascicle may have muscle fibers, okay. So, if you take a fascicle that also has a few, maybe up to 200 muscle fibers, a few hundred of them, muscle fibers, and each fascicle is covered by the perimysium. So, you have the bunch of fascicles covered by the epimysium, then each fascicle covered by a perimysium. So, these coverings are important. The perimysium, that is where a lot of the blood vessels, the nerves go in to the muscle. So, these are like the junctions, these tissues that are covering these fibers, they provide the pathways for the blood vessels, for the nerves, signals, etc., to reach the contractile element of the muscle. And all of these are also provide elasticity to the muscle, okay. So, that is the role of the perimysium and the epimysium. Then So, the fibers, if you look at it, they are covered by the endomysium. Again, which carry more of the capillaries, the smaller blood vessels and the nerves to innervate the muscle. So, these are also very cylindrical and they are thread like, okay, and they are fairly long also, like they are depends, but they could be 15 to 30 centimeter long, and they are kind of and they generate the force. So, in the muscle fiber you have thread like structures called the myofibrils. Okay. Okay. So, the myofibrils make up the fiber, about 80% of the fiber is made up of these myofibrils. Okay. And if you look closely at a myofibril, then you see the structure that is responsible for the contraction of the muscle. So, you have what are known as filaments in this myofiber. So, you have some thick filaments. So, okay. This. You have these are thin filaments called, let me erase this. You have some thick filaments and thin filaments. Now, these can kind of move relative to each other. Okay. So, this, the thick filament is called the myosin, that is your myosin, and the thin filament is called the actin. Okay. So, you have the actin and the myosin. Okay. So, we started off at the level of compartments. And then we came to compartments are groups of muscles. Muscles are groups of fascicles. Okay. Fascicles are groups of fibers. Fibers are groups of myofibrils. And myofibrils have these filaments. Okay. So, these are like a series of stacked together, these filaments. So, one filament is like what I showed you here. This is one filament and they are kind of stacked like that. So, this is the structure that of the muscles. So, 1 unit filaments, 1 unit is 1 unit of myosin plus actin is called the sarcomere. So, that is considered the fundamental unit for the muscle. Okay.
What is the difference between sarcomere and myofibrils?
So, myofibrils is this filament structure, bunch of the sarcomeres. Okay. The individual unit in the myofibril is a sarcomere.
Stacked along the way.
Yeah. So, longer a muscle, you have more of these sarcomeres stacked, and so you can probably guess what happens in the contraction, right? It is like a sliding action that takes place. It is called the sliding filament theory. So, when you have more of these stacked, right, you get more, that is shortening, and that is why the longer fibers can contract more than the shorter ones in the peniform arrangement. Okay. Okay. So, this is a better picture of the myosin and actin filaments. I will not go into the chemistry of it or how it does the chemical action that causes it, but essentially what it does is when there is a stimulation, okay, the myosin filaments have like these heads. Okay, if you look closely at the myosin filaments, they have these flexible heads, and they basically act as hooks onto the, so the heads have a flexible region, they hook onto the actin filament, form like a cross bridge, that is what it is called, and then pull it, and then relax again and do that again. Okay. So, when they are activated, it hooks on, pulls it like a rowing action. It does this, then back, then again another site, it latches onto and pulls. So, this stays where it is. It pulls the actin filaments closer together to close this gap. This H zone that you have, that is the gap that can be closed by this action. That is what is causing the, that is what is your contraction. Contraction is basically closing of that gap. So, in the contracted state, the actin filaments basically move closer together. Okay. So, this is called the sliding filament theory. This is the most commonly used explanation for muscle contraction. It was proposed by Huxley, and so the thin actin elements so the projections, let me just hold, I will just use a new page. Projections on the myosin form cross bridges with the actin and during contraction the thin actin elements slide towards each other. So, in the contracted state, filaments kind of overlap. And the amount of force developed is proportional to the number of cross bridges that are formed. So, even when a muscle is not moving, right, like when it is not, if it is, when we say if it is contracting, it means the cross bridges are holding on to one another. Okay. There may not be movement between the two, like and we will talk about the different types of muscle action later. We call the isometric action, right, when there is no movement, like if you are standing still and nothing is really moving, but your muscles are contracting, or I could contract my muscles without causing movement. Okay. That is called an isometric contraction where there is no movement. So, my muscles are acting, muscles are contracting, and the force that is produced is, even though there is no movement, the cross bridges are being formed, and that is what is resisting, that is what is producing the tension in the muscle. Okay. So, when a muscle is given is given a stimulus, so it will contract and then relax. This is called a twitch. Okay. For it to develop force, these twitches happen. So, one twitch it has to build one upon the other. Okay. And then, so for instance, it will go like this. So, a bunch of twitches together form a somewhat steady force, but when it finally reaches that steady force production, okay, that is called tetanus. So, when the twitches build upon one another to maintain that force, then the muscle is said to be contracting tetanically. So, this limit is called the tetanus, and the muscle is said to be contracting tetanically. Okay. So, the nerves have to send this signals and they have to kind of build upon one another to reach this steady limit. Here it is called an unfused tetanus. So, these are terms you will come across, muscle tetanic muscle contraction. So, you will know what it means, when the signals, when the frequency of the signals is such that it reaches that steady force production, that is your tetanus. Because this initially in a twitch, this is your contraction period, and then that is your relaxation period. Okay. So, this is your relaxation. So, before it fully relaxes, you give it another twitch, and then that is how it builds up. So, if you look at the length tension relationship for a sarcomere, so at that level, okay, then they have done experiments like this and looked at, so if you look at on the x axis you have percentage of the sarcomere length. So, 100% means it is at its resting length. Okay. If it is less than that, then it is shorter than its resting length. So, if it is at 60%, it is at a 60% of its decreased length. That is what the x axis tells you here. Then you look at, so you look at the tetanic force production, okay, at these different lengths of the sarcomere. So, if the muscle is and the tension is expressed in terms of the maximum tension that can be produced by that sarcomere. So, this here is your 100% of the tension that the muscle is capable of producing. You find that it is capable of producing the maximum tension close to its resting length, right. That is where it is capable of producing maximum tension in the muscle. If it is shorter than that, then you can see here, okay, the actin elements are kind of overlapped, and that interferes with, so the number of cross bridges are affected by this kind of overlapping of the actin elements, and so the force production falls off steeply. Similarly, if the sarcomere, if the muscle contracts when it is lengthened, okay, so something else is lengthening it, and then you apply a signal to contract the muscle, then not enough cross bridges can be formed. So, again the force drops off when it is beyond the resting length. So, under the resting length, beyond the resting length, your force production falls off quite dramatically. Close to your resting length is where you have the best force production in the muscle, because the cross bridge formation is maximized in that
in anatomical position that we in the diagram.
So, we are now looking at the sarcomere, we are looking at the microscopic level, but we will come, yeah, you
At that position, then person should feel its muscles should have a high tension in the muscles.
If you contract them, you can contract them.
But it's in the natural state we are getting the maximum forces from this curve at 100% of length.
The
It's
maximum tension at the in the anatomical position. Okay, that may or may not be the case, because if I look at the biceps for example, I am in the stretched position here. I am not in the resting position for the biceps brachii, right? So it's not necessary that, so the anatomical position, that's why it's only like a reference position. Okay, it's a reference position for us engineers to talk about relative positions and movements and to define. So it's basically for that. Yes, Ajay.
This implies at the resting position, I can apply 100% of the.
Yes, you can apply if the muscle is capable, that is the maximum tension the muscle is capable of supporting.
That is if I
Apply.
begin from the resting position.
At the muscle's resting position, so you have the sarcomere, okay? It's capable of generating some maximum tension. That maximum tension will happen close to its resting position. It cannot exert, it cannot generate tension greater than that amount, okay? So if you change the length, it's only going to go down from there. That's what this means, okay? So this is the behavior of the muscle, the contractile element of the muscle alone, the sarcomere. So when we talk about this length tension relationship here at the sarcomere level, we are talking about only the contractile component. But you know that there is a lot of connective tissue also involved in this whole structure. So the whole muscle behavior will also depend on behavior of the surrounding connective tissue as well as the tendons. And how the tendons behave, okay? And I think we will talk more about that in the next class, because we are almost out of time, okay?